Let's welcome Dr. Shanna Swan, epidemiologist and professor, Department of Environmental Medicine and Public Health, Icahn School of Medicine at Mount Sinai, New York City.
Hello. Today I'm going to tell you about a problem that I've been working on for the past 20 years. I'm going to start by describing the problem, and I'll talk about its causes, its consequences, and some solutions. So let's start. The problem was first discussed in 1992, and it was announced by this paper that came out of Copenhagen, which announced that there had been a genuine decline in semen quality over the past 50 years. This was really dramatic, and there was a lot of pushback on this paper. Many skeptics suggested that this was actually due to various kinds of bias and confounding. I was one of the skeptics. And so I spent 6 months trying to make this trend go away by considering things like whether the method of counting sperm had changed over the study period, or maybe the kinds of men that were included, or their BMI, their smoking, and so on and so forth. And I spent 6 months collecting all these data from the 61 underlying studies. And when I was all done and I did the analysis, nothing had changed, not to the first decimal place. So at that point, I had to stop and say, maybe it really is true that sperm count has declined. And if so, maybe the environment is playing a role because it's too fast to be genetic. So I spent the following 20 years almost, since 1992, trying to figure this out. And part of that work was to conduct a meta-analysis, which we published in 2017. When this appeared, it went viral. Unlike the first paper in 1992, which was not given much attention, this one was given huge attention. And what we showed in that paper was that for samples collected between 1973 and 2011, sperm concentration had declined 52.4% in Western countries. So at the start of the collection period, there was almost 100 million sperm per milliliter. But by 2011, this was less than half. This was very dramatic, and it didn't seem to be slowing down when we looked at the last 40, 30, 20, or 10 years. So at that point, I began to take it seriously. And as I looked across the literature, what I realized was that there were reproductive problems that were getting worse in men and women. And for men, it wasn't just low sperm count. There were other measures of genital development, including reduction in testosterone and birth defects. And fertility overall was declining. And in females, there were problems with such as miscarriage and early puberty and so on and so forth. And when you looked at the rates at which these things were changing, surprisingly, they were all going south, if you will, at about 1% per year. So I call this the 1% effect. So what could be causing this dramatic decline in reproductive health in so many areas? So when I think about causes, I separate them into genetic and environment, as do many people. And as I mentioned, genetics is not a likely explanation because this is 2 generations, 50 years perhaps, and it's too fast for that. So that leaves environment, and you can break environment down again into chemical exposures and lifestyle factors. I'm not going to say much about lifestyle factors, but they are important. And it is true that our daily lives now being more stressful, our fact that we're more obese, the fact that we smoke, that we have a lot of alcohol consumption, and so on and so forth, are things that can affect reproductive function. But I'm going to focus on the chemical exposures. And among the classes of chemical exposures, I'm going to focus on those that have the ability to alter our body's natural hormones. And that's because I'm interested in reproduction, and reproduction depends on the steroid hormones. But among the class of chemicals that we could consider under EECs, we can include the phthalates, which I'm going to talk about most, the bisphenols, the perfluorinated compounds, pesticides, and flame retardants. And here are some examples illustrating these classes. So the nonstick pan illustrating the PFAS, the rubber duckies illustrating phthalates, flame retardants, a crop duster illustrating pesticides, tin can, bisphenols, and again, another exposure to phthalates, which is in personal care products. So my focus has been on testosterone, because I'm interested primarily in male reproductive function, and testosterone is key to that. So let's ask, what chemicals can interfere with testosterone? And the answer is phthalates. So phthalates are chemicals that are found in everyday products and found in our food, personal care products, shower curtains, and it might get into our food through something like a milking machine, which even though the milk is organic, the milk is collected through a tube, and that phthalates in that tube can leave the tube and enter the food and get into our bodies. Why is this important? Well, it's very important when the fetus is developing in utero. So if we think about early pregnancy, initially the sex organs are undifferentiated, the same in genetic males and females. Then with the action of testosterone in early pregnancy, there is differentiation to the male typical and female typical genitals. But if the testosterone is not present, then you get the female option. If the testosterone is present, you go in the male direction. And it turns out then when a rodent mom is exposed to phthalates, the expected rise in testosterone shown in the left-hand graph, which is very marked at a certain time in pregnancy, a very short window called the male programming window. Under the influence of phthalates, that rise is completely obliterated. That is, the mother exposed to phthalate has a male fetus who does not experience the testosterone surge. And from the prior slide, you know that that testosterone surge is necessary for the differentiation of the male genital tract. So what else can happen? Well, it turns out that when that mother is exposed to, for example, diethylhexyl phthalate, one of the most anti-androgenic or testosterone lowering phthalates, a lot of things happen. And these are so marked that they've been given the name the phthalate syndrome. So what goes into the phthalate syndrome? Well, this incomplete masculinization, which consists of a number of things, including smaller penis, possibly a malformed penis, undescended testicles, and then for me, most interestingly, a shortening of the whole perinatal area, which is called the anogenital distance. This is a measure that has been used in animal studies since 1912, I found a reference, and has been used in toxicity testing, but it had not been used in human toxicity testing until I started to ask, what about humans? We're exposed to phthalates. Do we experience the phthalate syndrome? So I designed a study to look at that. So what we did was we took the mother's urine, because that's how you know how much phthalate is in a person's body, to look at the urinary metabolites. And you get that measured at a lab like the Centers for Disease Control. And then you get the infant. Now, what will you look at in the infant? When we started doing that, we weren't sure, but we mimicked the rodent study and tried to see the same endpoints in this little male infant. So we looked at testicular descent, we looked at the penile width, we looked at and learned to measure repeatedly and reliably the anogenital distance, and we looked at scrotal size. And what we found was the phthalate syndrome in humans. We published this in 2005, and it was pretty big news, and it actually influenced legislation. Because this is the first time that a syndrome named after a class of chemicals had been identified in humans. And then we repeated the whole thing because that's what you have to do. You have to replicate things. And in 2015, we published that replication. So together with the animal studies and the in vitro studies and our human studies and subsequent human studies where this has been shown repeatedly, we can conclude that the phthalate syndrome phthalates cause the phthalate syndrome in human males. And it shows that phthalates are anti-androgenic, that they're endocrine disruptors. And we also showed in this and other studies that AGD actually is a very good marker of how much androgen the fetus sees in early pregnancy. But what does that have to do with sperm count? Well, it turns out these babies couldn't tell us that because they don't have sperm, but it turns out that AGD is a permanent marker. In rodents, at least, if you're small at birth, you're small in adulthood. If that's true in humans, and we're not sure of that, then we could look at an adult man and we could say, is his anogenital distance related to his sperm count? And the answer is yes. And that's what we showed in this study, which was done in Rochester college students. And we see the direct relationship, longer AGD, higher sperm concentration. Another study showed that this was related to fertility, infertility associated with shorter AGD. So together with across these studies, which took 20 years, we go from prenatal exposure to phthalates to the anogenital distance in the newborn, and then to decreased sperm concentration in the adult. So we've made a link from this class of chemicals, endocrine disrupting chemicals, to sperm. We also, by the way, looked more directly at, for example, pesticides in one of our studies where we showed that pesticide exposure in adults was related to lower sperm count. So it's not just prenatal exposure. But the timing for this is really important. Whether it's early or late makes a really, really big difference. And I'm going to demonstrate that with this slide. This is about smoking, not about phthalates or pesticides, but the story is the same. So when the man smokes around the time of conception, and that's important because the sperm that he's producing at that time will go on to conceive the pregnancy. If he smokes, his son has about a 40% reduction in his sperm count. If the mother smokes when she's pregnant, her son will have a 40% reduction in sperm count. And those changes are permanent. They're irreversible because they're changes that occur during the organization of the genital tract. But if the man smokes himself in adulthood, his sperm is also decreased, but maybe half as much. And if he stops smoking, the good news is his sperm count can recover. So depending on when the exposure is, you have a difference in magnitude of effect and a difference of permanence of effect. So what are some other consequences of this problem? One thing that's not very well known is that sperm count is not just a measure of reproductive health, it actually is a measure of lifetime health. And men with low sperm concentrations have higher rates of heart disease and obesity and reproductive cancers, and they actually die earlier. And that's true whether or not they have children. And by the way, I want to focus attention on the right-hand category of sperm counts of 20 to 39 million per milliliter. Those are below 40, as are all of those to the left of that. And when the sperm count drops below 40, then you can see these effects on longevity, and we also see the probability of conceiving in any cycle going down pretty quickly. So when sperm count goes below 40, we're in trouble. And I want to point out that in 2011, we were at 47 million per milliliter. That was 10 years ago, and the chances are very good that we are already close to this cutoff. It's not just humans. There are species all over the planet that are being exposed to these chemicals and are showing serious problems with their reproductive success, some of them even being endangered or even extinct. So we're not alone on this planet. These guys are not asking for this exposure. We're subjecting them to it. So what are some of the solutions to this problem? First of all, I think we need to recognize that very few chemicals have been tested directly. It's better in Europe, but it's not great there either. And many of these chemicals have been, quote, grandfathered in and never tested. So what is this testing that we want? We want testing to remove chemicals that are hormonally active, like the phthalates, like the bisphenols, like the pesticides, like the flame retardants. We want to remove the chemicals that cause harm at low doses because our exposures are low. And we know from multiple experiments that they do cause harm at low doses. We want to remove those that are environmentally persistent because we want them to be gone. Once they're gone from our bodies, we don't want them in our fat. We don't want them in our food. We don't want them in our soil. And we want to remove those that have not been tested. And we have to replace those. New chemicals have to be created because we have needs for these chemicals. And these chemicals have to be without endocrine disrupting effects. They should be free of low dose adverse effects and minimally environmentally persistent. And finally, they have to be tested. They have to be shown to be safe prior to marketing, which is not the case in this country, although there is REACH regulation in Europe that requires that. So we have to regulate by assessing low dose effects, mixture effects, environmental persistence, and risk in untested chemicals. So that's what I have to say today. If you want to read more about this, I suggest you read Countdown. This is the book that I wrote with Stacy Colino, which goes into great depth into this story. Lots of references, lots of resources. You can order it, and thank you for your attention.